Welcome. In this video, we're exploring how enzyme activity in cells is tightly regulated to maintain metabolic balance and adapt to changes in the environment. The mechanisms we'll cover include feedback inhibition, reversible covalent modification, proteolytic activation, and the use of zymogens. One of the most fundamental ways cells regulate enzyme activity is through feedback inhibition, also called end product inhibition. In this process, the final product of a metabolic pathway acts as an inhibitor for an enzyme earlier in the pathway. It's a classic example of negative feedback. When the end product accumulates, it binds allosterically to an enzyme in the pathway, altering the enzyme's shape and reducing its activity. This mechanism prevents overproduction, conserving energy and resources while maintaining homeostasis. Another critical regulatory mechanism is reversible covalent modification, with phosphorylation being the most common type. In phosphorylation, a kinase transfers a phosphate group from ATP to specific amino acid residues, such as serine, threonine, or tyrosine, on the enzyme. Phosphorylation can either activate or deactivate the enzyme, depending on its role in the pathway. Conversely, dephosphorylation, the removal of the phosphate group, is catalyzed by phosphatases, reversing the effects of phosphorylation. This form of regulation allows for rapid and reversible changes in enzyme activity in response to cellular signals, playing a vital role in processes like signal transduction and metabolic control. Some enzymes are synthesized in active form and require a proteolytic activation to become functional. This involves the irreversible cleavage of peptide bonds within the enzyme precursor, causing a conformational change that activates the enzyme. This mechanism is essential when a rapid and irreversible activation is necessary. For instance, digestive enzymes such as trypsin and chymotrypsin are produced as inactive precursors called zymogens in the pancreas. They are only activated by proteolytic cleavage in the small intestine, preventing premature activity that could harm pancreatic tissue. Speaking of zymogens, these are inactive enzyme precursors that require controlled activation. The conversion usually occurs in response to a physiological signal or specific environmental condition. A prime example is the blood clotting cascade, where zymogens are activated in sequence to ensure clotting occurs only when necessary. The transformation of fibrinogen into fibrin by thrombin exemplifies how zymogens are used for rapid and controlled responses. These regulatory strategies work together to ensure enzymes operate efficiently and adapt to the cell's needs. Feedback inhibition provides a quick response to prevent excess product accumulation. Reversible covalent modifications, like phosphorylation, offer dynamic control and proteolytic activation, and allow for rapid irreversible activation when needed. Understanding enzyme regulation is not just an academic exercise. It has real-world applications in fields like medicine, where many drugs are designed to modulate enzyme activity, and biotechnology, where enzymes are engineered for specific industrial processes. To recap, enzyme activity is finely regulated through mechanisms like feedback inhibition, reversible covalent modification, proteolytic activation, and zymogen activation. These processes ensure metabolic pathways are efficient and responsive to the cell's needs, maintaining balance and adaptability in a constantly changing environment. Eager to learn more about enzyme regulation and master biochemistry concepts for your exams or research? Check out our app, King of the Curve, for detailed explanations, practice questions, and interactive resources designed to make complex topics understandable. Thanks for watching. Don't forget to like, subscribe, and explore kot.me for more engaging content on biochemistry and beyond. See you in the next video.